This is the second in our muscle tissue video series. And here are our learning objectives for this video. Number one, let's name the functional unit of skeletal muscle. Remember that the functional unit of an organ is the smallest level of organization of that organ, which demonstrates the properties of the organ itself. So to say it differently, your biceps brachii is nothing but a whole bunch of these functional units that are kind of slapped together. So if we understand the way one of these functional units work, then we understand what happens with the biceps brachii as a whole. Number two, let's describe the sarcomere. Well, I just let the cat out of the bag. The functional unit of skeletal muscle is the sarcomere. So what is your muscle, your pecs or your abdominal muscles? It's nothing but a bunch of sarcomeres put together. So your next question then is, okay, well, what does a sarcomere look like? How does it work? And that's what we need to describe. Number three, name the components of the thick filaments. And the thick filament you'll see is part of the sarcomere. Name the components and the function of the thin filaments, which as you'll see is part of the sarcomere. Number five, in the sliding filament model, the what filament slides along the what filament. And I will tell you already, the thin filament slides along the thick filament. One more time, the thin filament slides along the thick filament. And so another thing, we could just actually title this, we could describe this as introducing the sarcomere, because that really is what our video is here, just introducing the sarcomere. Okay, so where are we at? So by now, hopefully you're a little bit sick of this picture. You've seen it so many times by now. And what we're looking at is this stuff over here. And so what calcium will do once it's released from the sarcoplasmic reticulum is that calcium will actually go to a sarcomere. Once it goes to a sarcomere, it'll activate the sliding filament process. So this thing right over here, what my cursor is going over, and you see like an orange strand, you see blue beads, and you see this yellow anchor. So this thing over here, that is your thin filament. This is another of your thin filaments. And then this thing over here, which is entirely in red with this thing sticking up, with this head sticking up, this is your thick filament. So again, we've got your thin filaments and the thick filaments. So in the big picture, what's going to happen is the thick filament will grab the thin filament. So if you look on this, see those little heads? We'll do more detail later, but those heads will grab. They're going to go up and they're going to grab onto the thin filaments, and they're going to yank them in this direction. And you see the direction of arrows here. So here they grab onto them and they yank them in this direction. And so that's why we say the thin filament slides along the thick filament. It's not the other way around. The thin filament slides along the thick. So this really is where we're at. So what's the sarcomere then? These things right over here are a very, very zoomed in, up close version at a section of the sarcomere. So let's zoom out and actually look at the sarcomere as a whole. So this right here is your skeletal muscle. If you look at that, this is an image under a microscope, and it looks like it's got these stripes. If you recall, we described this before in a tissues unit where we said that these are striped, or the fancy word was striated. So skeletal muscle is striated. And here it is again in a more kind of cartoon drawing picture, and you can see these stripes here. Well, where do those stripes come from? The stripes really come from the organization of these protein fibers. It comes from the organization of the thin and thick filaments, because they're not laid out just crazy and haphazard. They're laid out very precisely, and as they're laid out, when you take a step back, it forms these stripes. So now what we're going to do is that this right over here is actually an individual muscle fiber. And if we take that individual muscle fiber, I mean, it's been cut here and cut here, but this is one muscle cell that would come all the way through. So we cut open that muscle cell just to look at the inside, and what we're seeing is a whole bunch of these myofibrils. Well, what is a myofibril? They're little tubes of sarcomeres. So if you take that section of myofibril, it looks like a tube. Then, and I know I used the analogy of a McDonald's straw before to describe the muscle fiber. And so if this whole thing is a McDonald's straw, what you can think of is tiny little miniature straws shoved within that McDonald's straw. And so these tiny little miniature straws, sometimes they're sticking out of like these fancy drinks and stuff like that. And so these tiny little miniature straws are the myofibrils. And if we take a myofibril and we zoom it in, this is what we see. And so I'll tell you right off the bat that these thicker looking red stripes. See that red stripe that I'm outlining right over here? These red stripes here are the thick filaments, and all the blue thin guys over here are the thin filaments. Remember, of the thick and thin filaments. These are really, really important. You see that they're covered here in these learning objectives. So remember, the thin filament, this guy over here, the thin filament is going to slide along the thick filament. And how does it slide? The thick filament will grab it and slide it along the thick filament. And so that's what we're looking at right over here. These red, big, thick things are the thick filaments, and all the blue guys here are the thin filaments. Okay, Z-discs I'll talk about in a second. What I mean are the blue lines running in this direction. All right, so now if we zoom on that a little bit more, so where are we at? We're at one of the tiny little thin straws shoved inside the bigger McDonald's straw, which is the muscle cell. So these are the myofibrils shoved inside the muscle cell, and there's hundreds and hundreds of myofibrils within them. If we pull out a myofibril, what are we seeing? I'm just going to make up a number. In a myofibril, we're going to see 70 or 80 sarcomeres laid up end on end. So see this here? That's called a Z-disc. This is a Z-disc. Don't worry about what it's made of for now. So that's a Z-disc, and that's a Z-disc, and this is everything in between. All that stuff in between Z-discs, that is a sarcomere, and the sarcomere is the functional unit of the skeletal muscle. So this is the beginning of this sarcomere, and that is the end of this sarcomere. On the other hand, this right over here is the beginning of this sarcomere, in which the end has been cut off in this particular image. So if we take a little, if we zoom in a little bit more, here's what we're going to see. We see this thing. There's my Z-disc, and there's my Z-disc. And again, this unit right over here is your sarcomere. This right over here would be another sarcomere, and this right over here is another sarcomere. So what we're seeing here is one whole sarcomere, and then basically a half sarcomere that's been cut here, and a half sarcomere that's been cut right over there. And you see them laid out this way. The terms I-band, A-band, don't worry about those. H-zone, don't worry about those. I'll tell you very explicitly what you need to know. And what you need to know are Z-discs and M-lines. So Z-disc and M-line, thin and thick filaments. Okay, so if we look at this thing and we blow it up, here's what we see. There's my Z-disc and there's my Z-disc right over here. So what do you want to know? Well, the first thing, even if you have to just memorize it for now, is that what you want to know is that the Z-discs, number one, are the boundaries of the sarcomere. That's the first thing about them you want to know. And number two, you want to know that the Z-discs are where the thin filaments attach. So my cursor right now is tracing over a thin filament attached to the Z-disc. Here's another thin filament attached to the Z-disc. There's another one, and there's another one. And there's more thin filaments right over here attached to this Z-disc. So Z-discs serve as the anchoring point for thin filaments. So what do you want to know about the M-line? The M-line then serves as the anchoring point for the thick filament. And this is the thick filament right over here. So the M-line is where the thick filaments themselves are anchored. So now, what I want you to see, you know what, let's go to the next slide before I point this out to you. So if you go to the next slide, this is just highlighting this thing here. So when muscles contract, you already know, look, when your biceps contracts, your elbow flexes. So just think of your biceps for now. When your biceps contracts, the thin filaments slide along the thick. The thick filaments do the pulling, and the thin filaments then get pulled. And so what that means is that these guys, so as the thick filament reaches up, what it'll do is, I want you to picture this, it's going to reach up and it's going to grab this thin filament right over here, and it's going to pull that inward this way towards the M line. These thick filaments will grab these thin filaments, and it's going to pull that in this direction in towards the M line. Well, now what I want you to imagine is that what are the thin filaments attached to? The thin filaments are attached to the Z-discs. So what does that mean? It means that as each of these guys start pulling, as each of them starts pulling in towards the M line, then what's going to happen is this Z-disc over here is going to start migrating because they're attached to each other. That Z-disc starts migrating towards the M line, and this Z-disc also starts migrating in towards the M line. So that's what we see. So what happens from there? Now, the Z-disc isn't going to get all the way there. It's just going to move in that direction. So I'll say that this sarcomere, let's say from Z-disc to Z-disc, if we were to measure this out and call this 12 inches, obviously that's a fake number, then when this thing shortens, it shortens from 12 inches down to, let's say, 8 inches or so, okay? So we've reduced that length by a third. What did I just do? I shortened the sarcomere. I shortened the sarcomere. And I do that along all of these sarcomeres this one, this one, this one. All these guys are pulling. All these guys within the myofibrils, all these sarcomeres start shortening, and all of these guys collectively, if each one of them goes from 12 inches to 8 inches, what do we see then? We see shortening of the muscle. And when the muscle itself shortens, what does that allow to happen? It allows point A to move towards point B, or allows point B to move towards point A, which is the way that we explained muscles in the previous videos. So when a muscle contracts, what happens to it? It shortens. And why does it shorten? Because gazillions of sarcomeres laid end on end and top to bottom are shortening as their thick filaments are grabbing onto the thin filaments, yanking them in towards the M line, pulling along the Z discs. Okay, so we'll have a little bit more description of that in the next video, but that is the general layout for now. So let's see. Let's zoom in a little bit more. There's your M line here. There is your thick filaments, and there's a whole bunch of proteins in the thick filaments, but all of them are made up of only one type only, and that is called myosin. So let's talk about the thick filament. When you look at the thick filament, it's made up of this protein, which is called myosin. If you look at myosin, myosin's got three parts that I want you to know. It's got a tail, it's got these heads, and a flexible hinge region right over here. And what that means is the head can be positioned here or the head can be positioned here. Now, what is the thick filament? The thick filament is a whole bunch of these guys bundled together so that it looks like this, which you can imagine. All these tails are stuck together and all these heads then are sticking out. And that flexible hinge region allows that head to actually pivot. So this right here is the thick filament. So to get into that learning objectives, to go back to learning objectives, name the components of the thick filament. Well, it's actually just myosin. But then if I were to say describe myosin, which I don't have written up here, but I really should after that, I should say describe the parts of myosin. Then from there, you should say, well, myosin's got heads, tail, and a flexible hinge region. And the thick filament is nothing but a bunch of myosin slapped together. So there we have it. That is myosin. Recall where we've seen myosin before. We've seen it right over here. Remember that this is myosin? And this is showing you just a single flexible hinge region with that head in this position or in this position over here. This position versus this position. One more time, this position versus this position. A lot more on that in the follow-up video. So that's myosin. So the next portion then is, let's say we described the components of the thick filaments. Let's look at the components and the functions of the thin filament. The thin filament then, again, is this portion right over here, and the thin filament is what gets grabbed, right? The thick filament grabs the thin filament and yanks it inward. So remember that I said that, let's see, if we copy that, then what ends up happening is that it will come this way. So one Z-disc, this Z-disc will move in this direction as indicated by the arrow. The other Z-disc will move in this direction as indicated by the arrow, therefore shortening the sarcomere. So the thin filament, what are the components there? Well, there's actually three of them. When you look at the thin filament, those blue beads, those little blue beads are called actin. And the key thing about actin that you want to know is that it contains this darkened portion here, these darkened portions that if you zoom in, you can see pretty clearly here. And those darkened portions are called myosin head binding sites. They're called myosin head binding sites. So if we were to say actin, what do you want to know? They've got myosin head binding sites. So what does that mean? Well, that's what myosin binds to. Remember that I keep saying that myosin reaches up and grabs actin. So if we go back to that video, not the video, this part, myosin reaches up and grabs actin. Well, what does it grab onto? It grabs onto the myosin head binding site of actin, and that's that darkened portion. The next component of the thin filament is this long goldish brownish strand called tropomyosin. And what is tropomyosin? Tropomyosin, his function, tropomyosin is to cover and, when the time is right, to uncover the myosin, the myosin head binding sites. Okay, that's what tropomyosin does. So it covers over, and when the timing is right, it uncovers the myosin head binding sites. So you can see right now, if I were to ask you, tropomyosin, is it covering or uncovering the myosin head binding sites? If you look at that, I mean, it's sitting right on top of those darkened pits. So it looks to me like they're covered. Right now, the myosin head binding sites are covered. And when they are covered, myosin cannot bind to actin. One more time. When tropomyosin is covering the binding sites, myosin cannot bind onto actin. When it's uncovered, then myosin can bind onto actin, and once it binds onto actin, it can yank and pull like it was doing in this video, okay? In this picture, I should say. So here, if you look closely, tropomyosin is not covering over those darkened portions. So it is not covering up. So your next question then is that, well, when does tropomyosin know to uncover the myosin head binding sites? Well, it's a great question. And that happens because of our third component of the thin filament called the troponin complex. Troponin is a complex of proteins. Sometimes you'll just hear people casually say it's troponin protein. That's fine. Potato, patata. Complex just mean it's made up of different subunits. So the troponin complex, what does it do? So the troponin complex is that third and final portion of the thin filament. And what that does is that it does two things. And I'll do the less important role first. The first, which is the less important role, is that what it will do is that it will anchor tropomyosin onto actin. It just serves as a physical anchor. Great. Now what's the more interesting thing? It binds to calcium. And after it binds to calcium, what it does is that it then twists. And as a result of physically twisting, what it will do, it will slide tropomyosin away from the myosin head binding sites. That's what troponin really does. So again, imagine troponin here. He's got receptors for calcium, and when calcium binds to him, he does a little twist. And when he does a little twist, he actually slides tropomyosin off to the side. And that's what's going to initiate our sliding filament model. And so already that gives you a preview then for if you ever had been wondering what's up with calcium. We went through all this trouble with a series of videos on action potential simply to release calcium. Well, what's the point? Well, calcium binds to troponin. Well, who cares? Troponin then twists and drags away tropomyosin from the myosin head binding sites on the actin portion. Well, who cares about that? Well, now that those binding sites for myosin are revealed, myosin can actually bind. And when myosin actually binds, then this filament, that myosin can bind and drag the thin filament along the thick filament, which then is the sliding filament model. So in the sliding filament model, the thin filament slides along the thick filament. Okay, let me close out this video, and in the next one, we'll just dedicate it simply to the sliding filament model, and we'll take a look at this picture over here. And this really is the heart of everything now. So all that action potential, all that calcium stuff, all that explanation of thick and thin filaments, this is our bread and butter. This right here is what allows you to bring a cup of water up to your mouth. When the biceps brachii undergoes this process, this is what allows your elbow to flex.